Hello, today we're talking about cell respiration. So, what is cell respiration? Simply, cell respiration is the breaking down of organic molecules, like the catabolism of organic molecules, in order to produce ATP for the body. It occurs with the mitochondria. What three main steps of glucose cell resp, and where does each one go? Where does each go, or where does each happen? I'm not too sure. So the three main first step is glycolysis. Glycolysis occurs in the cytoplasm, and what does it do? It just makes pyruvate. Second step is the Krebs cycle. That's where pyruvate gets turned into, it's like pyruvate is used and then it combines and goes like decarboxylation, and then we get acetyl-CoA. Acetyl-CoA goes in with oxaloacetate to form citrate, and then it just goes through the citric or Krebs cycle. And the main function of that one is just to produce NADH and FADH2, just electron carriers. The third step is electron transport chain. That is where the electron is just used to power proton motive force to the intermembrane space, so that the protons will want to rush into the matrix, and then stuff will happen, so then it'll just generate ATP. So what are the mitochondria important for? They're important for ATP. They make ATP. They're important for cell resp. That's why they're important. I don't know what else to say. For major what is unique about the mitochondria and where does DNA come from? So all the mitochondrial DNA comes from the mother. What's unique about the mitochondrion? It divides independently via binary fission, so it has its own circular plasmid DNA. And what else? It contains circular DNA, divides independently, has its own ribosomes. That's the other one. Okay, that's good. For major mitochondrial anatomy, so we got the outer membrane, then we got the inner membrane, then we got the intermembrane space, which is in between the outer and inner, then we got the mitochondrial matrix, which is within the inner membrane. What are cristae? For what reason? So cristae are the folds of the mitochondrial matrix. For what reason? They increase the surface area, which I'll get into later, but increase the surface area so that you can have more electron transport chains present. Two types of ATP formation. There's substrate level phosphorylation, and then there's oxidative phosphorylation. I think that's the correct name. I'm not too sure if I butchered that. Substrate level phosphorylation, oxidative phosphorylation. So substrate level phosphorylation is when you, when enzyme catalyzed reaction, a phosphate goes to the ATP. Not ATP, ADP, I mean. So it's just enzyme catalyzed reaction, phosphate goes from a molecule to ADP to form ATP. The other form is oxidative phosphorylation. That is when what's going on? The formation of ATP using energy derived from reaction of an electron transport chain. Energy derived from the reaction of an electron. It's just using an electron transport chain. That's all. Okay. So what are the reactants of glycolysis? Well, we're going to put in ATP. We're going to put in NAD+, and we're going to put in do we put FADH? Do we put FAD in? FAD. Do we put oh no, we don't. We don't have FAD here. We only have NAD+, and then we have ATP. Okay, that makes sense. So, yeah. Reactants with glycolysis is ATP. ATP. Wait, ATP and ADP then? Oh wait, why is it, where's that one? So we're putting in 2 ATP. Okay, let's just talk about the net reaction. We get 2 ADP, 2 phosphates, 2 NAD+, plus a glucose. When we get it out, we get pyruvate, H2O, 2 ATP, and 2 NADH. Plus 2H+, I think. Wait, what? Okay, let me just do that again. So what is the net reactants with glycolysis? I'm not going to put it on the numbers because I'm not trying to know that. But I think it's just 2, 2, 2, 2, 2 for everything. We have 1 glucose, we have 2 ATP, and we have 2 NADH. No, not NADH, NAD+. Now, when we go to reactants, we get pyruvate, we get H2O, and we get ATP, and then we got NADH. Yes, okay. Does glycolysis require O2? No. How is ATP formed in glycolysis? That's just using substrate level phosphorylation. Purpose of glycolysis is to form pyruvate. 2 minutes of glycolysis. The first step is going from glucose 6-phosphate using hexokinase to make 1,6. Whoa, I'm getting that wrong. Hexokinase. Okay, so we're going from glucose to glucose 6-phosphate. Okay. So the reactant is glucose, the enzyme is hexokinase, the product is glucose 6-phosphate. Why? It makes the phosphate negative so it stays inside the cell. Yes, I remember that. The other step is going from fructose 6-phosphate, and then we're going to 1,6, like fructose 1,6 diphosphate or biphosphate. What does it? It uses PFK, phosphofructokinase. Why does it do it? I'm not sure. Why does it do that actually? I think the importance for this one is that this is the irreversible step. And then like that's just like what kind of maintains the like that's how like ATP production is regulated through inhibition. So if there's too much ATP, then PFK will stop doing its work. I didn't remember that one. Okay, under what pathway would these steps occur? This is like the aerobic, so it has oxygen. Where does pyruvate carboxylation occur? Where does it happen? In the mitochondria? Intermembrane space? Or takes place in the mitochondrial matrix? Yes, okay. What makes pyruvate decarboxylation possible? I believe it's just the coenzyme A. What does pyruvate decarboxylation make? It makes acetyl-CoA. How is ATP produced in a citric acid cycle? It uses substrate level phosphorylation. What is the very first step for the citric acid cycle? Wait, why do I have two questions here? What is the very first step for the citric acid cycle? So acetyl-CoA will combine with oxaloacetate to form citrate. Citrate, there will be 7 steps afterwards, and most of those are just used for forming NADH and FADH2. And it also makes GTP and also makes ATP, but GTP is basically just ATP, etc. So I don't know, I just say etc. as a filler word. How many times does citric acid cycle occur for each glucose? There's 2 times because the pyruvate is a 3-carbon molecule, so you have to do it twice. For every acetyl-CoA, what are the products? I don't remember this one, bro. I don't remember the exact numbers, but I remember it's FADH2, GTP, ATP. We also make oxaloacetate as well in the end. Okay, so it's saying 2 acetyl-CoA molecules. Oh wait, no, that's the outcome. Oh, we're just talking about pyruvate oxidation. Oh, so with pyruvate decarboxylation, we make 2 acetyl-CoA, 2 molecules of NADH, and 2 molecules of CO2. Now for the citric acid cycle, we make 6 NADH, 2 FADH, 2 ATP, 4 CO2. Where'd the GTP go? Maybe they just said it was unimportant. Oh, it's right there, bro. Yeah, it says 1 GTP. Just doesn't show up. 6 NADH. Okay, I don't remember the numbers, but maybe I should. Main purpose of the citric acid cycle is just to make electron carriers. What are electron carriers? They are... What are they? Enzymes? No, not enzymes. But the purpose is just to carry electrons. What happens to them? They get reduced because they gain electrons. It's a coenzyme nucleotide molecule. What happens to them during earlier processes? They just lose, they gain electrons, so they get reduced. Yeah. what is the electron transport chain? Where? What the electron transport chain is a chain of electron transports. Where does it happen? It's in the mitochondrial matrix on the inner membrane. How does the ETC work? It forms an electrochemical gradient. So what will happen is that the electron carriers will carry the high energy electron, drop it off at complex 1, electron will just drop it out, keep on going. So NADH goes to complex 1, FADH goes to complex 2. The electrons will just hop on over, just keep on going, and then they'll get caught. The electronics, I'll get into that later, but what happens like with those electrons getting power, like with those complexes getting powered, those active transporters will push hydrogen ions to the outside of the mitochondrial matrix into the intermembrane space. So then we'll have a electrochemical gradient. What is the process of the E minus considered? Process of proton pump. So the process of the electron chain going down, that's exergonic, and then the process of the proton pump is endergonic because using energy. So yeah, so like it's kind of like coupling those two reactions. What happens to the last electron? So it gets captured by oxygen. The final electron transport, like the final electron acceptor, is oxygen. Oxygen will get that electron and it'll form H2O. What is the term for this? Final electron collector. I don't know. Final electron capture. Final electron acceptor. I think I said that like at least one time. What protein in this makes ATP? It's the ATP synthase. What is this process considered? Oh, it's considered oxidative phosphorylation. What does the electrochemical gradient provides proton motive force? It's just force by protons. Oh wait, no, the movement of ions. I think that's called chemiosmosis. What process does the crystal provide? You get more surface area, more electron transport chains, more ATP. Key difference of anaerobic respiration to aerobic respiration. Anaerobic respiration is only without oxygen. Aerobic is with oxygen. What does fermentation include? It contains glycolysis. What is the main purpose of fermentation? To reproduce NAD+. Where does fermentation occur? In the cytoplasm, because that's where glycolysis occurs, right? So it will just happen in the cytoplasm. I'm not too sure. Let me check. Oh wait, okay, wait. I want to talk about anaerobic respiration versus aerobic respiration. Aerobic respiration is with oxygen, right? So the purpose oxygen has is to collect the electron at the end. But in anaerobic respiration, we don't have oxygen to collect that thing. So something else will collect that oxygen. So something like sulfur, something nitrogen. I don't really know. Something like that. What does fermentation include? There's two glycolysis. What is the main purpose? I already said that. Where does fermentation occur? That's what I was trying to find, but. I want to say it happens in the cytoplasm. Oh, for what accepts the electron is SO4 2 minus, NO3 2 minus, NO3 minus, or sulfur, not nitrogen. Damn, that was tough. Where does fermentation occur, bro? Well, let me search this up. I don't see it. Where does fermentation occur? In the cytoplasm. Cytosol. Okay. Two types of fermentation. There's lactic acid fermentation and alcohol fermentation. Which organisms do it? Says each one. So humans do lactic acid, like animals, animals. Then what organisms do alcohol fermentation? Yeast, plants. Steps to each one. So with lactic acid fermentation, there's no acetaldehyde, so there's no electron capture in the end. So what will happen is that pyruvate from glycolysis will come in, it will form NAD+. Okay, wait, I'm like kind of BSing myself right now. Pyruvate will go to NADH, and it will form lactate and NAD+. NAD+ is replenished and able to be utilized in glycolysis. Lactate is transported to the liver. Okay, I know that, but how does it happen? So pyruvate, 2-pyruvate plus 2-NADH from 2-NAD plus lactic acid. Lactic acid will go to the liver to complete the Cori cycle and form glucose. On the other hand, we have alcohol fermentation. That's when NADH will NADPH is for photosynthesis, right? So NADH will come in, it'll combine with acetaldehyde and it'll form alcohol and also leave acetaldehyde will become ethanol. Okay, wait, so pyruvate will become acetaldehyde. Wait. Pyruvate will go to acetaldehyde plus CO2, then acetaldehyde plus NADH. Okay, so wait, I'm not fully getting this wrong. So there's 2-pyruvate, that's a 3-carbon molecule. Then we got acetaldehyde, which is a 2-carbon molecule. So pyruvate will go to acetaldehyde, release the CO2 because that's where the extra carbon's going. And then from acetaldehyde we'll get NAD plus. We're gonna get NAD. We're gonna get NAD plus ethanol. So the acetaldehyde will become ethanol, while in lactic acid fermentation we're not gonna have any acetaldehyde or the middle thing. And it occurs in the cytoplasm. I found it. Does fermentation make ATP? No, it doesn't. No, it doesn't. Where does respiration occur in prokaryotes? It occurs on the cell membrane rather than the mitochondria, because they don't have membrane-bound organelles. Okay, what is the difference between metabolism and cell rest? Metabolism contains all of the anabolic and catabolic reactions. Cell rest is just like cell rest is catabolic, so it's like just like a part of it. What macromolecules other than glucose can be used? So we could use lipids, carbs, like longer chain carbs, and proteins. We can use nucleic acids. 4-carb metabolism pathways plus what does each do? Damn, bro. What does it mean by 4-carb metabolism pathways? Well, okay, bro, what am I even asking, bro? Okay, 4-carb metabolism pathways. Okay, so there's glycolysis, there's glycogenesis, and glyconeogenesis. That was rough. Let me find it. Glycogenesis. So glycogenesis is forming glycogen. Glycogenolysis is breaking down glycogen. Gluconeogenesis is forming glucose from different molecules, like different things, macromolecules, and then glycolysis is just the process of breaking down that glucose and making it into pyruvate, right? Yeah. So glycogenesis, that's going from, okay. What does each do? So glycogenesis forms glycogen from glucose, or glucose 6-phosphate. Glycogenolysis goes from glycogen to glucose 6-phosphate. Gluconeogenesis forms glucose or glucose 6-phosphate from other macromolecules. Glycolysis goes from glucose 6-phosphate to pyruvate, just forming pyruvate. So the crossroads here is the glucose 6-phosphate. What 2 ways to regulate carb metabolism plus 2 effects? There's insulin and glucagon. Insulin is when blood sugar is high and it tells the body to store sugars. Glucagon is when the blood is low, so you need to break down the stuff. So when you have insulin, you're going to want to break down. No, you're going to want to store it. So you're going to want to do glycogenesis and Wait, glycogenesis and glycolysis, right? Yeah, okay. So glycogen, okay, and then glucagon, you're gonna want to break it down. So you're gonna want to do, if you're desperate, you're gonna do gluconeogenesis, if you really need to, but then you're gonna inhibit glycogenesis, promote you're gonna prevent glycolysis because you want to store the sugar for important things, and then I think maybe the first one doesn't really consider what's the, there's like one, right? There's like, you're gonna prevent what am I gonna say? You're gonna prevent glycogenesis, you're gonna promote glycogenolysis. Oh, it says inhibit glycogenesis. Oh, yeah, you're gonna trigger glycogenolysis, inhibit glycogenesis and glycolysis. Okay. Damn, bro. What am I doing? Okay. To gain energy from a triglyceride, what must be done first? What is the process called and what is used? What do you have to do first to a triglyceride? You need to break it down to its components. Okay, yeah. To gain energy from a triglyceride, break it down into the components, and it's called lipolysis. What does each part create? Where do they enter? Bro. Okay, so with the triglyceride, we got 3 fatty acids and one glycerol. So the glycerol, what even happens to the glycerol, bro? I remember what happens to fatty acids. We'll just go with fatty acids. Fatty acids will go under beta oxidation. Wait, that's right? That's like something common knowledge. Beta oxidation. That's what I said, right? I hope. It occurs in the livers. Oh no. It does not happen in the liver. It happens in the mitochondrial matrix. So what does it create? Well, for every time, so what we're gonna have is like this hydrocarbon chain, right? And it's gonna break down. It's gonna cleave carbon off. Now, when we cleave off those 2 carbons, we're gonna make acetyl-CoA. And we're also gonna make an NADH or NAD+. We're gonna make 1 NADH and 1 FADH. Okay, so 2 carbons are broken off and form acetyl-CoA, and we form what? We form 1 FADH2 and 1 NADH2. And we just keep on going until there's nothing left. The glycerol, on the other hand, will get phosphorylated and turn into G3P. Okay, so that's all you need to know. Okay, let me restart. What does each part create? Glycerol will get phosphorylated and form G3P. Where do they enter? It'll enter into glycolysis because that's the intermediate of glycolysis. Fatty acids will get cleaved through beta oxidation. Every 2 carbons will form acetyl-CoA, and you also form 1 NADH and 1 FADH2. What is the preferred energy of the brain? Well, you want to use glucose. It's just readily available, easy to break down. Secondary source, lipids is easier because with lipids, you'll want to break down. With fatty acids, with lipids, it's harder to break down, but you get more energy. What are epidocytes? What are epidocytes? They break down lipids? No. They specialize in storing lipids, I think. This was a practice question that I got wrong. That I don't remember now. Epidocyte, bro, it didn't even. Epidocytes. Sites. Specialized cells storing energy as triglycerides, regulating metabolism, acting as an endocrine organ. Sheesh, that's tough. Okay, the more I know. Okay, break down proteins into what in order to be used in what? Paul, like, what are these questions, bro? So we break proteins down into like the amino group it has, the NH3, but that's not good to have ammonia, so we need to combine that with something and make urea. Urea gets pissed out. And I think that's the only thing we can use for that. I think it was a very short section, so. Oh wait, ketone metabolism. Secondary source is ketone. Ketone bodies are formed from fatty acids in the liver, then transported to other tissues. Can be funneled into the Krebs cycle, can be used as energy source in the heart and brain. Okay, so it's ketone, not, I mean, like, I guess it's lipids, but it should say ketones. That's better. Ketones or glucose. Proteins, least desirable. They're made up of amino acids. They need to get broken down. So oxidative deamination, remove the amino group. And then the ammonia gets converted to urea. In order to make the remaining ammonia converted to urea. Yeah, okay, that's all I think you need to know. What happens first? Where does it happen? Converted into what? Bro, what? What am I? Where does this happen? I don't know. It doesn't say anywhere here. Well, I don't know. Where does it happen? Converted into what? Urea? Oh, I think I said. Oh, you just have to break it down. Then you have to break it down. I think that's going to happen in the kidneys. And then convert it into what? It's converted into urea. So, yeah, that's all for now. I have to do another one for chemistry. Goodbye.